Hi, this is Alex from devstories.eu. AlphaFold, Protein Structure Prediction, Episode 2 of 21. You want to predict the physical shape of a single protein sequence, but looking at that sequence alone is not enough. To figure out how it folds, you actually need to examine its evolutionary family tree to spot synchronized mutations across millions of years. Understanding how an algorithm processes those evolutionary patterns is exactly what we cover in Inside AlphaFold 2, Architecture Overview. The architecture operates as a continuous flow of data, transforming a string of amino acid letters into a 3D shape. It begins with the target sequence. Before any neural network processing happens, AlphaFold searches massive biological databases to gather two specific inputs based on that sequence. The first input is the multiple sequence alignment, or MSA. This is a collection of similar protein sequences from other organisms. If two amino acids in a sequence constantly mutate together across different species, they are likely physically touching in the final folded structure. The second input consists of structural templates. These are known 3D structures of proteins that are highly similar to your target sequence. These raw MSAs and templates feed directly into the evolutionary layer. The evolutionary layer translates this biological data into two distinct mathematical formats that the neural network can process. These are the evolutionary mutation history and the pair representation. Once created, both representations enter the Evoformer stack. The Evoformer is the engine of AlphaFold 2, consisting of 48 distinct processing blocks. Here is the key insight. Inside each block, the MSA representation and the pair representation talk to each other. They exchange information to refine their respective data. If the evolutionary data in the MSA representation strongly suggests two amino acids interact, it updates the pair representation to pull them closer together on the abstract distance grid. Conversely, if the pair representation realizes that placing two amino acids together violates physical space constraints, it updates the MSA representation to reevaluate that evolutionary link. This cross-communication happens continuously as the data flows through all 48 blocks, producing a highly accurate map of internal relationships. People often assume the Evoformer generates the final physical shape, but it does not. The Evoformer only builds abstract mathematical representations of distances and evolutionary links. It outputs highly refined data matrices, not a physical object. To get the actual folded shape, the data leaves the 48th Evoformer block and enters the structure module. The structure module takes the refined pair representation and the original sequence data and translates that abstract grid into actual 3D atomic coordinates. It assigns an exact X, Y, and Z position in space for every atom in the protein backbone and its side chains. The success of AlphaFold 2 hinges on the Evoformer continuously forcing evolutionary history and spatial constraints to agree with one another before a single 3D coordinate is ever drawn. That is all for this one. Thanks for listening, and keep building.